Hi everyone. In today's experiment, you're going to learn a technique called subculturing. So all subculturing means is taking bacteria from a source like an agar plate and growing it somewhere else, culturing it somewhere else. So that's what subculturing is. And the purpose of subculturing is to study an individual species of bacteria. So for example, the reason we do this is you've all streaked bacteria on agar plates, and you'll see various colonies here, different colors, different shapes. So they're different species of bacteria. If I'm interested in doing further experiments on just one of the colonies, I want to subculture that. I want to grow it somewhere else so that that's a pure culture of my bacteria. So that's what we're going to do today. The material you're going to need for today's experiment is I want everyone to take their agar plate that they have bacteria growing on, that they grew from this week. So all of you have a plate that you have various colonies growing on. I want you to have in your lab kit, there's a Ziploc bag and it says preserve culture. There's 2 tubes in there. Take out the tube that doesn't have anything written on it, and it's just a slant of agar. So we call this a slant. That's what people call it. It's agar, but it's poured in a slant way so it solidifies like that. And then I want you to have a Sharpie, which is what we're going to label. You always want to label everything that you use. And then finally, I want everyone to take out an inoculating loop, and these inoculating loops are for the purpose of picking up your bacteria and subculturing them. Okay, so the first thing I'm going to do is make sure that you washed your hands before doing any experiments and that you've cleaned your surface. I'm going to label my slant, so I'm just going to put the date on it and my name, and I'm just going to write the source. So I'm going to pick out a colony from the plate that I streaked my car keys on. Okay, so now this is the experiment that we're going to do now. You're going to choose a plate. All of you did 2 plates. Choose a plate that has a good amount of colonies on it. And so I have a plate and it has colonies on it. And the colonies, some of them have started to grow mold, which is okay, but you want to avoid using those colonies. So for example, I'm going to show you this plate. I have so many plates here, and you can see kind of a black color and fuzziness. That's mold. Around you may see colonies that are nice and round and there's no mold. Try to pick those colonies. That's what you want to pick. If for any reason all your plate is molded, just do this as a practice for next time. But what I'm going to do is here, if you can see my plate, there's 2 colonies that both are black and they're molded, but there's one on the side that you can't see in the video that's just like a very tiny white black circle, the colony, that's millions of bacteria. So that's what I'm going to subculture. So again, the purpose of subculturing is choosing one of the colony, not all. I'm not going to go like that. And then just streaking it on this slant so that I grow just that species. So I make a pure culture. Okay, so this is what I'm going to do this experiment. I'm going to open my inoculating loop at the stick side. I don't want to touch the loop because this is where I'm going to pick bacteria. And so I have my loop, and what I'm going to do is I'm going to take my plate, I'm going to open it, and I'm just going to touch, barely scrape off my colony of bacteria. So my circle of bacteria, I'm just going to gently just scrape it off. Again, try to not choose the mold ones. So there I have it. And it's going to look like a very little amount, but just know that's millions of bacteria in that little circle. Okay, so I have my bacteria on my inoculating loop. Now I'm going to open my slant, and this is probably the hardest thing, is try not to poke the slant. So the slant is just very soft jello-like material. What I'm going to do is I'm going to go inside with my loop, and I'm just going to basically just streak the surface, getting my colony on there. Okay. So if you're not experienced, it's gonna seem a bit weird. A lot of times students will poke the agar. If you do, it's okay, you're learning, but try not to. What you're trying to do, so I already did it, I closed it, is take your inoculating loop, and if this is your agar, you're just, here's a new loop. This is not my one with my bacteria on. You're just going like that on the surface gently, no technique. You're just basically going like that to grow that bacteria. And now I'm going to take this tube and I'm gonna incubate it. I envision it's going to take about 24 to 48 hours to see growth, probably actually 48 hours since it's room temperature. So we're going to keep it out. We're going to wait until our bacteria grow, and you've subcultured your bacteria. So now if you were in a research lab, you could use that bacteria to do infection experiments. You can do various enzyme testing, metabolic testing. And now our goal is, once we have bacteria growing, we're going to go use that bacteria, your subculture, to do various fun microbiology experiments.